Good morning, students. Today we are going to discuss the sporophytic phase in bryophytes. This topic is for M.Sc.Sem III students of botany. We have already discussed the gametophytic phase of the bryophytes, which is the dominant phase in the life cycle. So let us discuss the development of sporophyte in bryophytes. After the act of fertilization, which takes place in water, the male gamete fuses with the female gamete and zygote is formed, which ends the gametophytic phase of bryophytes. The sporophytic phase in the life cycle comprises the zygote, embryo, and the sporogonium. Zygote, here the fusion of the sperm with the egg takes place, and this is the pioneer structure of the sporophytic phase. It secretes a wall around it and enlarges in size. The zygote is retained within the venter, where it immediately divides without undergoing any resting stage to give rise to the embryo. Now, the development of the embryo. The zygote lying in the venter undergoes repeated cell divisions, and the cell enlarge to form a spherical mass of undifferentiated cells, which is called as the embryo. It covers the venter cavity. The archegonium is persistent. The venter expands as a closed envelope over the developing embryo. So this forms the calyptra, and the neck of the archegonium later on withers away or dies away. Now let us begin with the union, the zygote. The first division of zygote is nearly a horizontal. It is in a plane at right angles to the long axis of the archegonium. The next division is at right angles to the first. The young embryo consists of the 4 cells. It is called as the quadrant stage. The figure C, you can see, this is the quadrant stage of the embryo. All the 4 quadrants are nearly equal. Each quadrant divides by a vertical wall at right angles to the preceding one, and the embryo at this stage consists of 8 nearly equal cells. Here only 4 cells are seen in longitudinal section. This is the octant stage. Succeeding divisions of the 8-celled embryos in irregular manner gives rise to a spherical mass of 20 to 40 undifferentiated colorless cells. Now the young sporogonium. Each of the outer cells of the undifferentiated spherical embryo divides by a periclinal wall. A single layer of the outer cells is separated from the inner layer of the central mass of cells. The outer one is called the amphithecium and the latter is called the endothecium. You can see figure D. The undifferentiated mass of cells having 2 walls. The inner one is the endothecium and the outer one is the amphithecium, and the whole of the the embryo is covered by the protective covering called the calyptra. The cells of the amphithecium are large and flat. They divide only by anticlinal walls to form the protective sterile jacket layer, which remains 1-celled thick and constitutes the wall of the young sporogonium. The cells of the endothecium are all same. They constitute the first cell generation of the sporogenous tissue, which is called as the archisporium. So undifferentiated mass of the amphithecium, the endothecium cells is called as the converts into the sporogenous cells. Now, as the sporogonium begins to mature, the archisporial cells divide and redivide to form a mass of sporogenous cells, and the last cell generation of the sporogenous cells functions as the potential spore mother cells or the sporocyte. The spore mother cells are the last structures of the sporophytic phase. During further development of the sporogonium, the sporocytes begin to separate and become more or less round. Their walls become mucilaginous and the contents become granular. All these sporocytes divide meiotically to form the spores. However, some of them disintegrate to form a nutritive fluid and are called the nurse cells. We believe that the nurse cells are the forerunners of the elaters in the advanced members of the group Marchantiales. We are taking Rickshaw as an example for the development of the sporophyte in the Bryophytes. As the spore mother cells enter upon meiotic division, their walls disintegrate, the wall of the sporogonium and the cells of the inner layer of the calyptra also disintegrate, and the rounded spore mother cells lie free in the ventral cavity, in the cavity of the sporogonium, surrounded by the outer layer of the calyptra. The developing spores are nourished by the nutritive fluids formed by the disintegration of some of the spores. The nutritive fluid is formed by the walls of the spore mother cells, non-functional spore mother cells, jacket layer of the sporogonium, and the inner layer of the two cells thick calyptra. All these structures give rise to the nutritive fluid which provides nutrition to the developing embryo. Now the act of sporogenesis takes place. What is sporogenesis? It is the transformation of the diploid protoplast of the sporocyte, that is the spore mother cell, into the haploid dispersal unit, the spores, with resistant walls. The meiosis division gives rise to the diploid nucleus of the spore mother cells undergoes two successive nuclear divisions and give rise to the spore mother cell or the sporocytes. Each cell of the tetrad now secretes a wall around it and is called as the spore. We can say as the spores mature, the spore coat thickens considerably and the spore consists of the exine and the entine. The exosporium, which is yellow at first, changes to orange and becomes black at maturity. The callose layer and the sheet of the spore mother cell, which encloses the four spores, disappear, and the spores separate and lie free in the cavity surrounded by the outer layer of the calyptra. Now, these are the stages of development of the sporophyte in some important members. What is the structure of the sporophyte in Rickshaw? Here, the zygote after fertilization increases in size and undergoes a single division, forming two-celled embryo. This embryo, by repeated divisions, forms 20 to 30 undifferentiated cells, which later on are differentiated into an outer sterile cell, the amphithecium, and an inner mass of fertile sporogenous cell, the endothecium. The amphithecium forms the single layered capsule wall, and the endothecium becomes the archisporium. Some of the potential spermata cells fail to produce spores. They become abortive and they give nutrition to they convert into the elaters. There is no foot and seta, and there is very little sterilization of the potentially fertile cells. It acts as a spore producing organ. So, the fertile tissue is more in the simplest member, Rixia. Now, regarding the sporophyte of Markensia, here the two-celled embryo divides at right angles and four-celled embryo is formed. The two lower cells, called the hypobasal cell, remains sterile and form the foot and the seta. The upper two cell, called the epibasal cell, is fertile and by repeated division forms upper part of the seta and the capsule. The capsule portion is differentiated into an outer amphithecium and an inner endothecium. The single layered capsular wall is formed from the amphithecium. The entire mass of endothecium acts as a sporogenous tissue and becomes the archisporium. Half of the archisporium remains sterile. Cells elongate, develop spirally called thickened band and become the elaters. The elaters are hygroscopic and help in the spore dispersal. The remaining half of the archisporium become the spore mother cell. Some of the sporogenous cells at the top are converted into sterile apical cell. So, the sporophyte of Markensia consists of a broad foot, well-developed seta, and the capsule. Single layer jacket layer of the capsule, sterile apical cap at the apex, and spirally thickened elongated elaters. This illustrates a step further in the progressive sterilization of the sporophyte with consequent elaboration. Now, the sporophyte of the Palea. Here, the hypobasal cell does not take part in the development of the sporophyte. The epibasal cell divides and differentiates into outer amphithecium and inner endothecium. The amphithecium forms the capsule wall and the endothecium acts as the archisporium. The upper half of the epibasal cell forms the capsule and the lower half forms the foot and the seta. The sporogenous tissue of the endothecium forms the spore mother cells and the sterile elaters. The elaters are long, slender, with double spiral thickenings on their walls. Some of the fertile cells become compact and form elatophores. This is used in spore dispersal. The sterilization further is advanced, forming the efficient spore dispersal structure, which is the elatophore. Now, the sporophyte of the Anthoceros. The sporophyte of the Anthoceros or the group Anthocerotopsida is quite complex and advanced. The two-celled embryo is converted to 8-celled embryo arranged in two tiers of 4 cells each. The lower tier, by further division, forms the massive foot, and the cells of the upper tier also by repeated division forms the capsule. The capsule in young condition differentiates into the outer amphithecium and the inner endothecium. Here the difference comes. The endothecium is sterile and forms the central column of the sterile cells called the columella. Surrounding and overarching the top of the columella there is a sheet of sporogenous tissue, the archisporium, which is extremely reduced. The archisporium forms the spore mother cells. The covering layer of the capsule and archisporium are derived from the amphithecium. So, till now, the endothecium was giving rise to the capsule or the spores, but in the members of the anthocytales, the amphithecium gives rise to the archisporial tissue. The jacket layer consists of the chloroplast and the stomata. This enables the sporophyte to lead an independent existence. This type of sporophyte shows marked reduction in the sporogenous tissue due to the more sterilization of the archisporial tissues. And the anthoceros, the members of the anthoceros have a long-lived sporophyte due to the meristematic region present in place of the seta, which divide and redivide to produce the archisporial tissue. Now, the sporophyte of the notothallus sporophyte is more or less similar to the sporophyte of the anthoceros. This notothallus we will be taking individually in the coming lectures. Now, the structure of the sporogonium in the sphagnum. Here, the body of the sporogonium consists of a large bulbous foot, a neckline, inconspicuous seta, and a nearly spherical capsule. The capsule wall is several layered thick. The outer layer is heavily cuticularized, which bears the several rudimentary functionless stomata. The inner cell layers are thin walled and compactly arranged. The epidermis forms a ring-like annulus towards the apical region of the capsule, delimiting disc-shaped operculum form the capsule body. The capsule has a central columella overarched by a dome-shaped spore sac that contains the haploid spores. The whole of the sporogonium is shifted out of the perichaetum due to the prolongation of the archigonial branch, the pseudopodium. The swollen sac-like distal end of the pseudopodium encloses foot of the sporogonium and is called the vaginula. Now, the sporophyte of the last member, the most advanced member, which is the funaria. Now, the sporophyte of the funaria. Here, the capsule of the sporophyte is developed from the epibasal cell, and the foot and seta from the hypobasal cell. The epibasal cell divides periclinally, forming the outer amphithecium and inner endothecium. The amphithecium becomes differentiated into the epidermis, the photosynthetic tissue of the capsule wall, and the outer spore sac. The amphithecium at the apex of the capsule forms the multi-layered operculum, while the innermost portion forms the peristome. The endothecium is sterile. The thin outermost layer forms the archisporium. The capsule is pear-shaped structure with the basal cell sterile and known as the apophysis. The outermost layer is the epidermal layer, below which the massive parenchyma cells with chloroplast are present. There are large air spaces traversed by delicate strands of parenchymatous cells known as trabeculae. Surrounding the lid of the capsule, a ring of cuticularized epidermal cells known as the annulus is present, which help in the spore dispersal. The progressive sterilization of the potentially sporogenous tissue is increased here. So, this is the extreme limit funaria, where the progressive sterilization ends. So this figure shows the stages of development of the sporophyte in some important genera of the Bryophytes. And here these are the mature sporophytes of some of the members of Rixia, Markensia, Palea, Anthoceros, Notothalus, Sphagnum, and Funaria. And the upper one shows the successive stages of the sporophyte development in the group Bryophyta. Now, the spore dispersal in Bryophytes. The mature sporogonium undergoes certain mechanical changes leading to its dehiscence, and the spores are dispersed from the parent plant. There are some structures present inside the sporogonium which help in this spore dispersal process. The spores are the first stage of the gametophytic generation that are formed from the spore mother cell by the reduction or the meiotic division. The mechanism of spore dispersal is of taxonomic importance to the Bryophytes. After dispersal, the spores germinate and form the new thallus. The continuity of the races is maintained by this structure. The different types of spore dispersal observed in Bryophytes are as follows. In Rixia, there is no special method of spore dispersal because the seta is absent, and the spore dispersal in this genus, as the spore bearing structure is very simple, the sporophyte is embedded in the gametophyte, and after maturation of the sporophyte, the spores are not immediately liberated, they remain inside the thallus. When the sporophytic layer undergoes decay, the spores are set free. In Markensia, the hygroscopic nature of the elaters present in the capsule helps in spore dispersal in Markensials in general. The capsule walls have ring-like thicknings. When the sporophyte becomes mature, the wall shows splitting at the periphery of the apical cap. In this way, the cap becomes detached and the remaining portion of the wall splits irregularly into 4 to 8 teeth-like structures. Due to the sudden elongation of the seta, the wall of the mature capsule splits longitudinally from apex to middle. The masses of spores are loose in state, and the elaters help their dispersal in different directions. In Palea, the mature capsule splits into 4 longitudinal wells. The hygroscopic movements of the elaters help no flicking away of the spores to different directions. Anthoceros. The spore dispersal process is dependent on the water content of the medium. When there is loss of water, the sporogonium becomes black or brown, then it is ready for dehiscence. The entire structure shrinks and the capsule wall strikes with the pseudo-elaters. At this stage, there appears a dehiscence line on the tip of the capsule. Gradually, it widens and enlarges downwards. The internal structures, that is the spores and the pseudoelaters, respond due to water loss and create a tension leading to the split of the capsule in 4 halves. The pseudoelaters help in distributing the spores outside. Funaria. Here the peristomal teeth help in the dispersal of the spores. When the sporogony matures, the columella and other internal tissues break. The spore sac is filled with spores due to this breaking. The thin wall holding the operculum gets detached and the peristomal teeth become exposed. The outer layer of the peristomal teeth are hygroscopic in nature. It bends out the cavity of the sporogonium, dries up, and gives a jerk, lifting out the spores outside. The seta also helps in spore dispersal. So, This is the mechanism of spore dispersal in the important genre of Bryophytes. Students, with this, I finish with the general introduction of the Bryophytes, and in our coming lectures, we will be taking the individual genera which are mentioned in your syllabus. Thank you.